Okay, so it's 9 o'clock, so let's get started. So welcome everyone to today's Immunotherapy seminar. Always, I want to thank our speaker, Dr. Jun Wang, to spend hours this late with us, give us a lecture, and obviously everyone online to be with us as always. So it's really my pleasure to welcome today's speaker, Dr. Jun Wang from NYU. So Jun currently is assistant professor in Department of Pathology, NYU School of Medicine. Jun actually complete his undergraduate study in bioengineering in Nanjing Tech University. And then further, he continued to pursue his PhD training in molecular immunology in Institute of Biophysics, Chinese Academy of Science in Beijing. After which he decided to continue his scientific training as a postdoc. So he moved to US in Yale Department of Immunobiology with Dr. Lieping Chen. After a very productive period of a postdoc training, he started his independent research program in NYU, as I just mentioned. So the main interested in Jun's lab is in the discovery and functional characterization of novel cell surface regulatory pathways as potential targets for immunotherapy of cancer, autoimmunity, and then different other diseases. So as I just mentioned, Jun did a very, very productive PhD and postdoc training. Previously, he actually explored the machinery of NT4-1BB, which is CD137 in liver toxicity, and then identify myelocell associated targets for reducing NT4-1BB immuno-related adverse effects. Further, he also characterized functional novel receptor ligand interactions from different novel screening strategy at the human genome scale, and was the first to discover LAX3, cyclic 15, and membrane-associated antigen presentation inhibitors as important tumor immune invasion mechanisms and new targets for cancer immunotherapy. So during the pandemic, in response to COVID-19, Jun's lab actually proposed model for COVID-19 immunopathogenesis and then discover normal myelocell receptor for SARS-CoV-2 as potential pathogenic mechanism and targets for virus-induced myelocell hyperactivation. So with really remarkable record in postdoc training and as an independent faculty, Jun was awarded as Immuno-Oncology Young Investigator Research Scientist Award, Team Science Award from Melanoma Research Alliance. And in 2021, he was awarded as V Scholar Award, Mark Foundation Aspire Awards. In 2022, he was awarded as BMS MRA Young Investigator Awards from Melanoma Research Alliance. So I think Jun's gonna tell us more about his recent work on the evasion mechanism for cancer. So thank you so much, Jun, to share your science with us. We are very much looking forward to your talk.
All right, thank you so much, Chuan, for the very thorough introduction, and thank you for hosting. So basically, I've been listening to Immunozone for over 2 years now. I think that's very informative. I've been very looking into this platform to communicate with the colleagues and potential scientists. So it's my great honor to be here. As Chuan mentioned, I've been focused on immunotherapy, especially for cancer. My interest is to identify novel evasion mechanisms. So today I'm going to share with you our one of the new projects working on cancer-related immunoevasion on class 1. So this is a disclosure. So a little bit introduction on myself, as Chuan just mentioned. So I was trained by Dr. Lieping Chen and also Shen Dian from Chinese Educated Sciences. So I got both PhD and postdoc training under Dr. Chen. So early time, especially when I started my PhD, I was very interested in. At that time, 2004, nobody really believed PD-1 can really become a drug for cancer. So that's why I started my PhD working on 4-1BB. This is a molecule that Lieping discovered in cancer. At that time, actually, 4-1BB shows very interesting, anti-tumor efficacy in the tumors. So, and the BMS actually initiated the clinical trial in the cancer. So, however, I was one of the very first scientists to identify 4-1BB have a liver tox. I discovered the mechanisms, and then I moved to United States around 2009. So actually, around the same time, PD-1 trial came out. 4-1BB had liver tox, but PD-1 have basically very successful and very little side effects. So that was one of the reasons why I decided to move into immune checkpoints, especially novel T cell checkpoints. So I started to work on LAG3, C15, still work on T cells, but T cell right now is very busy. So many checkpoints has been discovered, TIGIT, LAG3, TIM3. So given I started my lab around 4 years ago at NYU. So right after I started my lab, COVID came. So that's why I tried to understand how COVID modulates myeloid cells, especially how COVID interact with myeloid cells. So that's why we identified several receptors on the myeloid cells. At that time, starting from that moment, I was getting interested in myeloid cells. So that's why I've been thinking about what kind of myeloid biology is important for tumors. That's why we have this story. So in general, I have the both training for autoimmunity and cancer. And that is also the focus for my lab to building novel immunotherapies for both cancer and autoimmune disease. So the story came from the 4-1BB, as I just mentioned. 4-1BB is the molecule is really the inducible costimulatory molecule for CD8 T cells. And actually 4-1BB intracellular domain has been fused to CAR Ts, has been clinically approved for CAR T cells, because this is one of the most important molecule to boost T cell responses, especially for T cell memory. So around 1997, 25 years ago, actually we just wrote a paper, review paper with Lieping and Lacho Minero, who discovered this 4-1BB in cancer. So 25 years ago, it was discovered to be very interesting molecules can be a target to treat cancer. So, and afterwards, Lieping also discovered PD-L1. So when we start mouse models, tested all these anti-PD-L1 or anti-4-1BB, in most cases, we actually found 4-1BB is better than PD-L1 as monotherapies. As a combination, definitely it works also very well. So that is one of the reasons why many companies right now, they are testing PD-L1, 4-1BB bispecific. And also they're thinking about to combine PD-1 or PD-L1 with many costimulatory immunotherapies. So my PhD at that time was focusing on how 4-1BB can break T cell tolerance in the liver models. But actually we have HBV transgenic mice. For this mice, the liver, especially hepatocyte, is very susceptible for liver injury. So with this model, we found 4-1BB treatment actually induced liver injuries, showing by ALTs. This is an enzyme produced by hepatocyte. And later time, actually, you will find this model where the mice were having some kind of fibrosis-like syndromes. And ultimately, the mice goes to liver cancer. So this phenomenon is actually very similar to later liver tox showing in the BMS clinical trials of 4-1BB. And actually, around 2009, this trial was stopped because 2 patients died. So again, at our time, 2009, 4-1BB trial was stopped. And actually, 4-1BB trial, the first month when I came to Hopkins, showing very interesting efficacy in the human trials. So I was wondering, because as I mentioned, 4-1BB works so good in mouse models, but in a human, why it behave very different? Why PD-1 works poorly in the mouse, but in the human is so different? So I was trying to understand what we can learn from anti-PD-1 or PD-1 immunotherapies. As everybody knows PD-1 right now, PD-L1 right now is so successful. So I was thinking about maybe the key is coming from this type of feedbacks, because PD-1 positive, because first of all, PD-1 is very specific on T cells. And the T cell, normally naive T cell doesn't have PD-1, and the T cell goes to the tumors, become PD-1 high. And this T cell will secrete lots of interferon, and this interferon will induce PD-1 in the tumor site. to be said, it's like you have a T cell in the tumor sites, and they start to kill the tumors, and the tumor sense these interferon responses and upregulate PD-L1. And in these cases, it will suppress T cell. This is kind of negative feedback response. So there's another ligand called PD-L2 doesn't have that kind of feedback in the tumors. So that's why we think, right now PD-1, PD-L1 become the drug, not PD-L2. So we think the major principle, first, we have to target tumor site, more targeting disease lesion. Second is we have target tumor-induced immune feedbacks, not just like other immune checkpoints. It's more like immune feedback response. Third is we should have some kind of mechanism to repair T cell responses by targeted therapies. So that's why for myself, I've been thinking about to identify something beyond PD-1, and then whether this can enhance, responses of immunotherapies because right now only 20% of patients have a response to PD-1, PD-L1. So I think about other disease or other targets. That is the focus of my lab. So my lab right now is focused on cold tumors because many people know, PD-1 have a response, especially in the PD-L1 positive and also TO positive population. Basically, you have T cell, you have PD-L1, and then PD-1 targeting have a response. So for PD-L1 lective, still you have T cells for this type of molecules, this type of patients. So we're thinking about some other checkpoint like LAG3, C15, but still you have another population basically have very little T cells. That is basically the major problem for immunotherapy right now. What kind of molecules, what kind of targets can be there? So we're thinking about myeloid cells, especially because myeloid cell is the key molecules to modulate T cells, to modulate immune responses. So my lab has been focusing on novel targets. So as I mentioned, we're interested in novel feedbacks. For example, when we discovered FGL1, this is a novel ligand for LAG3. This molecule, why I think it is interesting, because normally produced by livers, but actually can be amplified by cancer cells. Now we know cancer cells, especially lung tumors, there's some kind of tumor hypoxia, oncogenic mutation where induce FGL1. And this type of feedback will shut down T cell responses through LAG3. So recently, last year, BMS antibodies has been approved, and LAG3 has been approved as a combinational therapy with PD-1. This antibody actually block both FGL1 and Cas2. So that's why BMS is very interested in my studies. So they hired me as their advisor. So another type of feedback, for example, C15, why I was interested in this molecule is because C15 normally induced by MCSF. This is the molecule induced immunosuppressive M2 macrophages. And C15 actually, this molecule is, if induced, will suppress T cells. And we have some new data showing C15 can modulate myeloid cells as well. Again, this is another type of feedback, not by interferon, by another type of cytokines, MCSF induced. This type of cytokine is opposite from interferon. That's why we think this type of feedback is different cohorts of patients. So for myeloid cell, now this is why we get into myeloid cells when I start my lab, because of COVID. We think myeloid cell is essential, cell type to initiate T cell responses, B cell responses, and even myeloid cell is important for tumor immunity and also for many of the immunological disease. So when I start to think about myeloid cells, especially when we think about the current landscape for cancer immunotherapies, as you will see, interferon has been improved, IL-2 has been improved, cytokine, those type of cytokine therapies, and also PD-1 therapy, LAG-3 therapies, the most of those molecules, targeted therapies are targeting T cells. There are actually very little therapies, many target myeloid cells. So that's why we need more myeloid cell targeted therapies. We need more basic biology understanding of myeloid cells. So the problem for myeloid cell is, first of all, CSF1 has been testing for a long time. This is one of the molecules drive macrophage, especially M2 macrophage differentiation. So there are two types of CSF1 blockade or CSF1 receptor blockade, or even small molecule target those molecules, and then targeted cell type populations. Those type of population will be depleted by antibody blockade. So the problem for this type of depletion therapy is there might have some kind of compensatory macrophage compensation. When you deplete this macrophage, you have other macrophage population coming back, can do even much more immunosuppressive job. So that's a problem. When you deplete macrophage, you have another macrophages to suppress T cells, to suppress immune responses. So CD47 has been very hot. The major problem here is whether macrophage-mediated killing of live tumors, that biology is important for tumor immunity. So many people still have doubts. So maybe this is one of the reasons why CD47 is not that interesting and promising at this point, but could be interesting in the future. So that's why for my lab, we've been thinking about, first of all, how tumor engage with myeloid cells, and especially how this myeloid cell will ultimately affect T cell responses. For example, myeloid activation and M2 differentiation or antigen presentation, especially antigen presentation. Because as we know, antigen presentation is very important. So first of all, antigen presentation, especially MSC class 1 binds to TCR, T cell activation, secrete lots of cytokines, recruit T cell, and even recruit lots of myeloid cell. This will cause inflammation, and this is a major cause of hot tumors. In cold tumors, because no T cell involved, and then as a result, very little inflammation can be triggered. So that is so-called cold tumors. So many cold tumors, especially another name is, there are two types of cold tumors. One is immune excluded. So T cells is around the tumor, and the second is immune desert. Basically, you cannot find any T cell at all. So both tumor types, actually, people have found no class 1 is kind of features. So people have been thinking about this question, class 1 no. I mean, everybody knows class 1 no in tumors. But when we look into the literature, actually one by Yale colleague, he was leading in this field. He was thinking about the major problem for class 1 downregulation was just because of the deficiency of the positive regulator. For example, B2M loss, this is one of the factors important for MSC class 1 machinery, or interference signaling. This is one of the signaling pathway trigger MSC class 1 expression. So it's because the loss of this positive signal that was generally this tumor field think about why class 1 has been downregulated in the tumors. But the problem here is, especially for this paper, if you look hard, looking into the evidence of, for example, B2M loss in the tumors, actually for this paper, they have screened hundreds of patients. They only found several patients have that kind of mutation. And even for that patient, not every tumor cell can be positive. So the question here is, what is a major cause of antigen presentation class 1 loss? Major mechanism. So what's a major mechanism? So I think still not very clear. But however, when I started looking into viruses, especially when I look into many literatures from SARS-CoV-2, from HIV, HBV, you'll find many of those mechanisms starting from the whole process of antigen presentation. For example, you have antigen process in the cytosol. Some viruses can shut down this protein degradation. And there are also some kind of mechanism can shut down antigen transport, the type 1, type 2, and also loading, and also trafficking, and also stabilization in the cell surface. There are actually some viruses on the cell surface. For example, EBV, there is some kind of GPCR, BILF1. This molecule actually binds to MSC class 1, shut down class 1 by internalization. There's another type of molecules. For example, this is a single transmembrane domain proteins. You will bind to class 1, actually compete with B2M. So those are mechanisms from the viruses from all levels of antigen presentation to shut down class 1. So the question here is, viruses has been so efficient to shut down T cell responses, has been so efficient to evade immune system. So cancer is also very smart, right? They should have some kind of mechanisms, something like that, especially in the cell surface. We already have evidence this type of mechanism can be on the cell surface, potentially can be antibody targets. So that's why at our time, the first day when I came to NYU, I was thinking about this question. Maybe tumors can actively evade antigen presentation through some kind of similar mechanisms. If those mechanisms exist, maybe you can use those as a potential cancer targets, therapeutic targets. So I work with Xu Feng, one of the very talented postdoc in my at NYU, actually from Dr. Infanti's lab. So we talked together, we wanted to collaborate with each other. So that's why we designed a screening system. So basically we want to, build a very sensitive screen system. So we fuse the antigen, for example, AFP in the human or OVA in the mouse. We fuse with a fluorescence. And then this antigen will be presented by HLA-A2. And then you can see the peptides, specific peptide. So we actually get a very specific antibody from Eureka. So Dr. Liu Chen, his company in San Francisco. So they built the specific antibody can detect this complex. So in the mouse, OVA can be presented by HLA-A2, and also this specific complex already there's antibody commercially available. So we found this system is, we definitely tried to titrate it a lot. So we found this system is very sensitive. Basically, BFP is kind of indicator of antigen amount, and another y-axis you can use antibody to detect antigen presentation, either from the mouse system, human system, and mouse system. Basically, all of those looks like linear association. So more antigen, more presentation, more antibody staining. So this looks like a very sensitive system. So with this system, we wanted to do whole genome scale CRISPR screening. Again, this was the first screen for the antigen and an MHC class 1 specific interaction for the CRISPR screen. This was the first screen. So we basically think about the high expression, low expression. If we infect the whole genome CRISPR libraries, we wanted to know what's happening, what kind of genes specifically is reached in either directions. So we, after thorough investigation, so we actually found many molecules important as a positive regulator. For example, type 1, type 2, those has been suggested to be important for antigen presentation. Those are top hits as a positive regulators. Again, those molecules has been suggested to be very important. So those type of data really validate our system is a very sensitive system. We can pick up low-end genes, although those genes are positive regulators. In particular, we're interested in some kind of molecular regulators. So we constantly see some molecules from both human screen and the mouse screen. So we wanted to know whether those molecules can be potentially important. So first of all, we built a whole network. So this is positive regulators. So most of the positive regulators, type 1, type 2, B2M has been well studied. But again, we still see many new genes from the positive regulators. Many of those genes are important for mTOR signaling, mitochondrial functions, and many of those genes are actually not very clear. So some of those genes definitely worth to study. So we built that work. So if you're interested, you can look into, and if you have any questions, we can discuss for further. But we are more interested in negative regulators. For these directions, I would say most of the genes are not clear at all. So that's why we're interested to know how we can decode the function and whether the relevance of those genes to antigen presentation. So because we did a screen for the antigen presentation, especially antigen specific peptides and MHC class 1. So we also want to know whether those genes are not just like specific, like whether those are general to all the HLAs, because in human there are HLA A, B, and C. So you also did another screen, which is general HLA A, B, Cs. So we did overlay. Actually, we wanted to know whether are there any more general molecules control antigen specific complex, PMSC complex, and also general MSC class 1. For general MSC class 1, actually on the surface already loaded with different type of peptides. So for this purpose is we want to identify something general. So we found 21 genes ranked by all three screens. This is the positive regulators, and also we identified some negative regulators. We found 44 common regulators. So if you really look at those molecules, and then you have some kind of common regulators, and also some genes specifically only exist in the PMSC screening. So the 44 genes on the left basically is a antigen specific screen plus the general HLA screen. So those are more general. So we're also interested in some kind of surface molecules, right? So we actually found on the left more general gene sets. We found more antigen, more membrane proteins. So especially the top three genes, SUS6, TME127, and ARP10. Then we are getting more interested. So it's like this type of general modulators are cell surface molecules, are they really having function to affect MSC class 1 directly because they are on the cell surface? So that's a basic hypothesis. So we also did some validation with the in vivo screen, basically to testing whether those molecules are related to T cells, related to and whether those molecules are related to tumor growth in vitro. So we want to identify something not just like affect tumor biology itself, it's really affects some kind of T cell biology. So with all kind of screen, we narrow down and we have the top hits. Basically, it's called SUS6. So this molecules is very specific on NK, myeloid, and the B cells. If you look at the tissues, basically this molecule is very myeloid related and also immune related. So these molecules is a single transmembrane domain proteins. As I mentioned to you earlier, for the mouse screening, sorry, for the virus screen, actually there's a molecule, single transmembrane protein binds to MSC class 1, that is from the viruses. So we also find a single transmembrane domain proteins, potentially shut down MSC class 1. So these molecules, as our time we don't have antibodies, so we made a flag, we can detect this on the cell surface. So this data suggests this is really a cell surface proteins. So then we want to know whether this molecule really regulate antigen presentation. So we did more validation. For example, in the mouse, this is the THP1 cells. It's more like a mouse, human myeloid cells. And actually, it's this type of AML, acute myeloid lymphoma cells. So we knocked it out SUS6. So we actually found more antigen presentation, AFP presented by HLA2. And actually, general HLA2, we also see some upregulation. In the mouse system, same thing. We found the KB ova and also general HLA can be presented, can be expressed in a higher fashion. So basically, validation. So another data is we want to know whether the modulation of these molecules will affect T cells. So this is the mouse system. We have an antigen, specific antigen ova, presented by H2AKB. And then we have a specific T cells, B3Z, and upon regulation, you will see IL-2. So if I knock it out as SU6, we see basically threefold enhancement of T cell activity, which is quite impressive. So for the killing, the same thing. We found more killing, and also we validated with the human system with the same antigen-specific T cell responses. We found the same thing in the human and the mouse. So then question here is whether this molecule will affect T cell biology and ultimately affect tumors. So we start with mouse acute myeloid lymphoma models. So with this model, we locked it out, these molecules, and then we see tumors actually slow down tumor didn't grow very well compared to the controls. So as shown by tumor volume, survival, and because this is very T cell specific. So we depleted the mice with CD8 T cells, and we found no difference. Basically, this phenomenon is T cell dependent, especially CD8 T cells. So we also tested some association. For example, the expression of SU6 associated with highly expressed in AML, and also have some association with the worse survival of the patients, and the expression of this molecule associated with T cell, selectively associated with T cell activation. All those are supporting data showing these molecules correlates selectively with T cell signature, with the survival, with the survival of the patients. But then the question here is whether this molecules is a general mechanism in all cancers. So that's why we studied many solid tumors. Those molecules are amplified in the PDAC. PDAC is very cold tumors, pancreatic cancer. Also, in other type of tumors, especially brain tumors, GBM, LGG are very high. All of those tumors are cold tumors as well. So then we tested many tumor cell lines. In the mouse cell lines, we tested over 10 actually. And the majority of those tumors, as you see here, if you look at all these molecules, you will see antigen presentation, especially class 1 expression, get enhanced. So it seems like a general mechanism, not just like blood tumors, solid tumor, majority of the cell line, if you really push to test the function, it does have some effect. So we tested solid tumor models. It works the same to shut down tumors, and also associated with we tried two different cell lines, B16, CD26, and also all of those are T cell dependent. If you deplete T cells, there was no difference. So at the same time, we tested many immunological responses. So small tumors, small tumor weights, in the tumor you will find more CD8 T cells, more CD8 functions. Those are, because you already have tumor difference, so you can predict you have more responses. So that's why for the first conclusion, so we have started a CRISPR screen, which are from in vitro and in vivo. We identify key players of tumor antigen presentation. And also we have built the whole network, positive and negative network. If you are interested, you can look into our list and maybe can be helpful for your tumor studies. And the second is we found this SUSD6 single transmembrane proteins depletion enhanced tumor antigen presentation and sensitized cancer cell to CD8 T cell mediated cytotoxicities. And this is effective in both blood tumors and solid cancers. It seems like general mechanisms. But the question here is, what's a mechanism? What's a real mechanisms? So first of all, we look at the key positive regulators of antigen presentation, whether locked out of these genes. We didn't find too much evidence in the transcription levels. So for the MR levels, we didn't find too much difference of HLA, B2M, type 1, type 2, all those important genes, important key genes as a positive regulators for antigen presentation. So we think this is not through the MR level. We also tested translation level, not too much difference for the global translation. But for the protein level, we did find knocked out SU6, you find more HLA, more B2M. It seems like protein level has been enhanced. So then question here is maybe affect stability of the proteins, right? So then we tested 4th degree and 37th degree. We didn't find too much difference in the 4th degree, but we do find difference in the 37th degree, suggests related to protein stability. Then the question here is like whether internalization, phagocytosis, more detailed studies. So we tried translation inhibitors, basically suggest this is related to translation or stability. We also tested more protein degradation. So this is an inhibitor which suppress lysosomal degradation. So for the control, when we give the inhibitor, MSC class 1 will be enhanced, meaning MSC class 1 will be very sensitive for lysosomal degradation. However, when we knocked out these molecules, there was not too much difference. So meaning SU6 is mainly control lysosomal degradation. And for proteasomal degradation, we also have an inhibitor, but we didn't find too much difference. So then we think maybe the major control of class 1 is lysosomal degradation. So then question here is how single molecules, SU6, can control MSC class 1 lysosomal degradation. Are there direct interaction or is there any other molecule can be involved? So by looking at the top hits, we found the TMEN127 and the WWP2. And then we look into literature. Actually, there was one study from a group in UK. So they were study pathogens. So they actually found TMEN127 associated with WWP2, and those two molecules itself cannot engage with class 2. But however, when there was some kind of pathogenic, some kind of pathogen, especially salmonella infection, there is a protein from the salmonella. This protein is a two transmembrane proteins, can really bridge class 2 with TMEM and then shut down class 2. So basically, they found a protein from the pathogen and then can behave like a bridger to form 3-molecule complex and shut down MSC class 2. So then the question here to us is, maybe SUS6 behaves some kind of adapter like STAT-D can bridge class 1 with WWP2. So in order to prove that hypothesis, we first look at the association of TMEN127, SU6, or SU6 with WWP2 in all the tumor TCGA cohorts. As you know, TCGA contains many tumor types, contain many tumor samples. We did find positive association of these 3 molecules. So this could be very hard, right? Because this is a big cohort of studies, many cancers. We found all these genes associated with each other positively in all cancers, ML, CLAD, many other types, cancer types. So this data suggests it may be very possible that all these genes can associate with each other and potentially, just like this paper suggests, can become a big complex. So we just tested with a very simple Western blot to see whether these three molecules can be together. So with Western, we found that those molecules actually can form potentially bind to each other. But Western is also not very clear, right? Many people don't believe Western. So that's why we did another study, which is split luciferase. So if you have protein A or protein B carries a large protein, large beta, small beta part of luciferase, if they have the interaction, it will bridge both part of luciferase proteins together and form luciferase. So basically, if you see the signal, might suggest there was a protein interactions. So we tested the interaction of HLA2 with S or T or this type of two molecules. So we found SU6 binds to MSC class 1 very strong compared to TMEM, but TMEM still have some interaction. So basically, HLA2 will associate with these two cell surface protein, but with S most strong. So then question here is, how about this E3 ligase, WWP2? So we found this E3 ligase binds to SU6 also very strong. So basically SU6 is the strongest proteins to engage with those molecules. And at the same time, another interesting data is for WWP2 binding with HLA, we didn't find the binding in the beginning, right? WWP2 doesn't bind to HLA in the beginning, but however, if we add SU6, still low binding, add TME127, still low binding, until we find two protein together, add into the system, and then HLA2 will bind to WWP2. So this is very interesting data, suggests E3 ligase binds to HLA2. Normally it doesn't happen until you have two molecules together. So with this complex, so if we knock it out SU6, so we actually found the ubiquitination of HLA2 has been reduced. So all of this data really suggests the E3 ligase WWP2 is involved for HLA ubiquitination and the stability of these molecules. So this came out as a model. So basically, we think class 1 itself will not engage with WWP2 E3 ligase, and these two molecules will breach MSC class 2. So then question here is why E3 ligase can be associated with these two molecules. What's the structural, what's the biochemical interaction mode? This is something we're working on. But our model here is E3 ligase will be bridged by these two molecules to the MSC class 1, and then mediate degradation of the molecules. So so you might ask me what's a application, right? So on one side, we're thinking about the therapeutic application. Second side is, are those molecules can be useful to predict as a biomarker to predict clinical outcomes, especially these three molecules, three complex. So we call STM, S, SU6, T is TME127, and W is WWP2. Can we really use this three gene signature to predict immunotherapy responses? So the answer is yes. So in the ML, we did see upregulation of these three genes. In the PDAC, this is a pancreatic cancer, we see upregulation of these genes associated with survival, looks very well to predict the response of the patients. So also we tested another molecule, TME127, we lock it down, we see the same thing to control blood tumors, to control solid tumors. Is blood tumors, the solid tumors, the same thing. So basically, target S protein or TME127, both have very similar phenotype. So another interesting data is we found genetic evidence. So recently, this data has not been published, but we found the TMEM knockout mice. So we found this molecule also highly expressed on myeloid cells, but compared to SU6, it's less specific. But however, we found we profile the phenotypes, we actually found knockout mice have especially for the B cell, T cell, all the immune cells, you found more class 1. For the myeloid cell, also more class 1. Basically, the major difference is most of the immune phenotype, most of the immune cells, you have more class 1. And if you look at the CD8 T cells here, the knockout mice have more CD8 T cells, but less NK cells. So the major phenotype of this knockout is you have more class 1, more CD8 T cells, but less NK responses. So that's why this data suggests this molecule is really a key modulator of a class 1 because of genetic evidence. Because of class 1, you might have more CD8 T cells. Because of class 1, you have less NKs because NK can be suppressed by class 1. So we also have a SU6 knockout. We also knock out SU6 in the myeloid cell. You also see class 1 because before we're just targeting this molecule in the tumors. In the myeloid cell, same thing. And in this knockout, we also found the same phenotype. All these myeloid cells, or especially myeloid cell, have more class 1, very similar to TME127 knockout. All those knockouts have more class 1, have more CD8 T cells. Phenotype are so similar. So until right now, so we think we are very positive to say what we found is a more general regulatory pathway, not just like for tumors. It can be a general pathway for both tumors and also extrinsically for myeloid, for T cells, for immune system. So this is something general regulatory pathway for class 1. So with that, I think the conclusion here is we found a membrane-associated inhibitory access with these 3 molecules, which binds to E3 ligase. Potentially, we also find another molecule, STAP1, can do a similar job. And the target this complex will lead to MSC class 1 degradation. If we target S protein or T proteins, or even in the future, we can target WWP2 E3 ligase. It will enhance MSC class 1, enhance immunosurveillance, and it is general pathway because not just tumor cells, myeloid cell, T cells, if we knock out this molecule, we enhance class 1. So that's why we think it might represent a potential targets for immunotherapy. And also, it might represent a potential biomarker for immunotherapy responses. So with that, I have to thank my my postdoc, Qiao Lu, and also another postdoc, Xu Feng. Both are co-first authors from, and Xu Feng is from the Advantis lab at NYU. So this was a great collaboration. And also my collaborator from Uraka Therapeutics, Dr. Liu, and also Peter from Yale, and also Patricia from UT San Antonio, and Dr. Zhang Kai from Yale. Thank my collaborators, and also thank my lab, thank my fundings. So thanks everybody, and then we can discuss if you have any further questions.